पोदार सर लिंक दिलाय मला तुम्ही.
बोला सर.
चॅटवर कोण लक्ष
लिंक
ठेवणार?
युट्यूबची
चॅटवर कोण
हा.
लक्ष ठेवणार आहे?
चालू
देता
केलं
काय?
की तीच आहे की
चालू-
तीच लिंक आहे. ते चालू करून बघा. ती सुरू करून बघा की आधी.
थांबा.
हम्म.
नाही ऑफलाईन आहे ते.
आता चालू दिसते. एक
लाईव्ह
मिनिट.
स्ट्रीम ऑफलाईन दिसते आता.
एक मिनिट.
मी लाईव्ह शेअर करतो सर. थांबा. ते नसताना मी लाईव्ह शेअर करतो.
थांबा लाईव्ह शेअर करतात थांबा. आत्ताची चालू केलं की
लिंक आहे.
लिंक वेगळे आले का?
हा मग ते सांग.
ही लिंक शेअर
आम्हाला
करायला
दुसरी
पाहिजे.
पाठवतो मग तुम्हाला.
पाठवा.
व्हॉट्सअपला
सगळ्यांना जायला पाहिजे
पाठवलं.
ती.
आता पाठवले
नवीन
का
लिंक सगळ्यांना
9:16
जायला पाहिजे. पोदार सर,
नाही ओपन होत. हा सर बोला.
ती नवीन लिंक सगळ्यांना
ही
जायला
झाली की
पाहिजे.
ओपन होते की लिंक?
नवीन लिंक
होते ओपन?
झाली की.
ओपन होते ना?
हा
दुसरी लिंक
झाली राऊत सरांनी सांगितलं.
हो
झालं
झाली.
YouTube.
नवीन लिंक ओपन झाली.
झाले ना? हा आता ती शेअर करा मग.
हो. आता आवाज आता म्यूट करा सगळ्यांनाच. पहिला सगळ्यांना म्यूट करा मग चालू मग शेअर करतो.
बर.
हो.
लिंक शेअर केलेली आहे. लिंक हॅज बीन शेअर्ड. सो बी अलर्ट. अमर एवढे पण आत आलंय.
Yeah.
Welcome. Now it will start in few minutes.
सांगा बोलू नका. युट्यूबच लाईव्ह दिसते आता. तुमचं काय किती मिळतं आपल्याला किती नाही YouTube वर. हे युट्यूबच आहे. कोण बोलतंय? बोलतंय? तो परत म्हटलं.
म्यूट करा, म्यूट करा.
नाही ते आता लाईव्ह YouTube नाही आता वेलकमची
युट्यूबच येते झूम येत नाही. हा?
वेळ झालाय म्हणा, चालू करायचं का नाही? एक सर चालू करायचं का नाही? हम्म. Am I audible? Am I audible, sir?
Yes.
At the outset, I pay my respectful homage to late Yashandraji Chavan Sahib, to Ventura Chavan, and Adarniya P.D. Patil Sahib. A very good morning to all. Hope you are staying safe in homes and enjoying a good health. Even though the world is locked down, it has also created opportunities like this one. On behalf of the Shivaji Education Society, Karad's Board for Higher Education, Yashwantharao Chavan College of Science, I welcome you all in this international webinar on Perspectives of Microbial Diseases 2020. Today we have with us our resource persons, Dr. Amar Evare from New York, USA, and Dr. Vaibhav Mahindarkar from Württemberg, Germany. Heartiest welcome to both of you. We have a proud feel that both the resource persons are alumni of this college. It is a matter of pride and honor that in this morning we have with us Honorable General Secretary of the Shivaji Shikshan Sanstha's Board for Higher Education, Honorable Prakash Patil sir, Bapu. I welcome you, sir. I also welcome Coordinator of Sanstha's, YB Chavan Computer Exam Guidance Center, Dr. B.N. Gopane sir. I also welcome our principal and chairman of today's webinar, Dr. S.B. Kengar sir, convener Dr. N.R. Sheikh sir, and organizing secretary, Professor T.B. Sawant. I also welcome the coordinators, Dr. Avinash Raut, Dr. Jaising Patil, and Shri Kishore Sutar. For next 2 hours or more, we will be listening and discussing the topic of current importance, perspectives of microbial diseases. No other word is required since the whole world is facing the pandemic due to a virus. Once again, I wholeheartedly welcome all the participants and resource persons in this virtual gathering. Now I request Principal Dr. S.B. Kengar sir for his introductory speech. Sir, please.
My humble tribute to Honorable Sri Yashwantharao Chavan Sahib, Honorable Sri P.D. Patil Sahib, Sau Vinutai Chavan. Good morning and welcome all in one-day international webinar on current perspectives on microbial diseases 2020, organized by Internal Quality Assurance Cell and Department of Microbiology, Yashwantharao Chavan College of Science, Karad. I feel honored to welcome Honorable Balasaheb Patil, Minister of Cooperation and Marketing, and Honorable Chairman of Sri Shivaji Education Society, Karad, Board for Higher Education, Honorable Prakash Pandurang Patil, General Secretary, Board for Higher Education, Sri Shivaji Education Society, Karad, Dr. B.N. Gopane sir, Coordinator YB Chavan Competitive Examination Center, and other office bearers. Warm and hearty welcome to Dr. Amar Evare, Postdoctoral Associate, Stony Brook University, New York, USA, and Dr. Vaibhav Mahindarkar, Württemberg, Germany. I'm here to give some about the college. Sri Shivaji Education Society, Karad, Board for Higher Education, founded by Honorable Sri Yashwantharao Chavan, former Deputy Prime Minister of India, established this college in June 1958 as a science college, Karad, with an objective of providing education in a science to students of Karad and surrounding area in Maharashtra. To keep his memory alive, the college was renamed after him in 1986. The college building, inaugurated in 1960 by then the President of India, Honorable Dr. Rajendra Prasad, is located in sprawling campus around 40 acres and has flourished under the able stewardship of Honorable Sri P.D. Patil Sahib. Now, the college successfully runs by Honorable Chairman Sri Balasaheb Patil, Cabinet Minister, Cooperation and Marketing, Maharashtra State. Today, Yeshwantrao Chavan College of Science, Karad, is one of the premier educational institutions in this region. This college is a granted institution affiliated to Shivaji University, Kolhapur. Our college is also an ISO 9001:2015 certified college and is now accredited B++ level by NAC in 2017 in third cycle. The college offers total 13 courses at the graduation BSc level. The college has also facility of postgraduate courses in microbiology and chemistry. College has recognized research laboratories for PhD in microbiology, zoology, and botany. MPhil courses in botany and zoology are also being offered in this college. 3 professional courses such as BSc in biotechnology, BSc in computer science, and BSc in computer science entire at graduation level. From this academic year, college has also started the MCA course recognized by YCMOU Nashik. The college has also received several grants for major and minor research projects and for infrastructural facilities from funding agencies like UGC, CPCB, and DST SERB. To achieve academic excellence, our meritorious students have consistently occupied top place in board and university merit list. In the academic year 2017-18, the college has received award of maximum number of merit scholarship in urban areas as well as in a science stream by Shivaji University, Kolhapur. Sanstha has also started YB Chavan Competitive Examination Coaching Center to provide a guidance to the students who come from the rural background, to develop good administrator, to create awareness among the students for various competitive examinations. In short, our Sanstha has the facility to educate students from KG to PG. So to this, I welcome you all in this international webinar on perspectives of microbial diseases 2020. Thank you.
Thank you, sir. Now I request Dr. N.R. Sheikh, sir, the convener of this webinar, to introduce today's first resource person, Dr. Amar Yaware. Sheikh, sir, please.
On behalf of Sri Yashwantrao, Sri Shivaji Education Society's Board for Higher Education, Yashwantrao Chavan College of Science, Karad, Department of Microbiology, I am introducing Honorable Speaker Dr. Amar Yaware for the first session of this one-day international webinar. COVID-19 has actually created a pandemic threat throughout the world, but we, the members of Sri Shivaji Education Society, Board for Higher Education, Department of Microbiology, are taking this as an opportunity to interact with you all people leisurely. So I'm trying to introduce Dr. Amar Yaware first. He has been a bachelor's and master's students in microbiology in Yashwantrao Chavan College of Science, Karad, which is affiliated to Shivaji University, Kolhapur. He was the best topper student of those days. He has completed his PhD degree at Academy of Scientific and Innovative Research, CSIR, NCL, Pune. Now, he is working as a postdoctoral associate at Stony Brook School of Medicine, New York, USA. He has research experience and is seen very much skilled and expertise in cell culture, biochemistry, molecular biology techniques, PCR, electrophoresis, cloning, expression analysis, and so many others. Till date, he has 12 research papers at his credit, of which 11 are in international peer-reviewed journals and one in national. He also has one national patent at his credit. He has attended so many national and international conferences. He has attended so many national and international conferences. He has been awarded by so many awards and honors. Some of the awards and honors are Best Research Scholar Award by NCL, RF Kirti Sangoram Memorial Endowment Award by NCL Research Foundation 2019. Then Best Oral Presentation Award, Best Paper Presentation Award, then Inspire Fellowship. Then he has been awarded a third prize for research article in College Magazine, and he ranked first in MSc Microbiology in all science subjects of Shivaji University, Kolhapur. Thank you. Thank you, one and all again.
Thank you, Dr. Sheikh, for the brief introduction of the first speaker. Now we are turning to the first session, and I request our today's first resource person, Dr. Amar Eware, for his talk. Dr. Amar, sir.
Can you allow me share screen?
Yes, please.
No, I'm not able to share a screen. Host, can you please allow me a share screen? I'm not able to share a screen.
Whether, sir?
Yeah. Thank you very much, Dr. Rena Sheikh, for a brief introduction, and Rekas sir and other convener of this seminar for inviting me to give this good lecture on this topic. Particularly, this is my first webinar, and it's midnight, so I hope it will be go smoothly. So let's start on this topic, particularly this latent tuberculosis and some aspect of the COVID-19. Today I will talk about just tuberculosis. What is the actual active and latent infection or disease, TB disease? What is the need of dormancy model and the drug screening assay to cure this tuberculosis? And I will also talk some aspect about the COVID-19 infection in a latent tuberculosis and how it's spreading, and what should be a precaution we need to take during this pandemic. So if we are going towards this TB prevalence worldwide, so TB is the one of the ancient disease, and as per WHO report, 1/4 of the world population is infected with the TB, and every new infection occur at about 1% of the world population each year. So it's very dangerous, and we need to treat it. If you look at the prevalence in the as worldwide, the more high risk or high TB incidence is found in the Middle East, African country, many African country, and most high risk is in the India, while the rest of Asia and the South American countries are intermediate risk, and the USA or the North America or the certain European country are on the low TB incidence. Keeping this mind, this figure will be tell about the will be joined with the COVID-19 infection, because the risk, if you find this high incidence rate is in South America, Africa, or particularly in India, and this region is started lately COVID-19 infection. So far, this tuberculosis is caused by the organism Mycobacterium tuberculosis. This is only a tuberculosis, means Mycobacterial tuberculosis species cause the tuberculosis, and another one species is recognized there, that is a Mycobacterium africanum, but that species is restricted to the some middle Africa countries, means a particular demographic countries. So it's not widely spread, and it's not that much shows the symptoms of the tuberculosis. If you look the symptoms of the tuberculosis, it shows the chronic cough with the blood containing a mucus, and along with that you will have a fever as well as night sweats and some kind of weight loss. So sometimes what happens, the fever or night sweats or the weight loss may be occur in other diseases, or it may be symptom of other infections, but the along with these symptoms you should have the chronic cough with the blood containing mucus. Then it will become confirms it will be showing the active TB infection. Otherwise it's difficult to identify the TB patient based upon the only this fever or night sweat or weight loss, and chronic cough should be there. But what happens if without these symptoms, most of infections may not have some symptoms, or it's remain asymptomatic, and in that case it's called as a latent TB, and the current rate is near about 90% population is suffered from the latent TB, and only 10% or 5 to 10% are showing a positive symptoms, and that are only active TB. And again that out of that 90%, 10% latent infection progress to the active disease due to the immune suppressive condition like as HIV or other co-infection whenever will be there in that particular patient. Then it will be caused the it will be reactivated and form the tuberculosis symptoms, active tuberculosis symptoms. Then actually what is the difference between the active and latent diseases, particularly active and latent tuberculosis diseases? The latent tuberculosis, TB bacilli infect but does not grow or multiply in the body, while this active TB diseases or that bacilli grow and multiply in the body. It's in a latent case, the person will feel person does not feel sick or not have any kind of symptoms, but in active TB the sick person will be sick and have many kind of means that earlier talk about that symptoms will be there. And this latent TB cannot spread from person to person because the almost all bacteria or the mycobacterial bacilli are remain in non-replicating stage. So it is not spread through the aerosol, but active infection will be spread. And in case of active, if it is not treated, then it will be cause death, while this TB can result towards the active diseases. So it's latent disease is always on the risk, so we need to diagnose it correctly. So whatever the available current diagnostic tests are the Montauk test, that is a tuberculin well-known test. Another is sputum culture, AFB, that is acid fast bacilli test, and some interferon gamma release assay. Gene expert, particularly PCR related technique. So you will be identify the genome of the mycobacteria in the sputum sample. And the chest X-ray. Chest X-ray come whenever will be there is a symptoms, then it will be helpful to identify or diagnose the actually active TB diseases. While there is no test particular for the latent tuberculosis, whatever these two tests are saying based upon the WHO report or WHO data, that is the World Health Organization, this test can be distinguished. If there is no symptoms and this test get positive, then it is considered as there will be a latent disease. But this is not true, because the tuberculin test is particularly get positive in the BCG infected patient, because and widely this BCG infected because this is a hypersensitivity reaction, so that if a person already immunized with the BCG, it will show positive tuberculin test, and so it's very difficult to diagnose the latent tuberculosis disease. And so there is a need of actual diagnostic of this latent tuberculosis. If you look about this TB pathogenesis, how it is actually cause the disease in active state or the latent stage. First upon this EMTB bacilli comes to the aerosol and infected to the alveolar macrophage, that is lung macrophage, and under the immune suppression condition, this bacilli multiply inside the phagosome, and it's not allowed generally to fuse with the lysosome, and this bacilli continuously multiply and cause the active disease. While if there is a strong immune response, this macrophage is recruit the other cells like as natural killer cell, other B lymphocyte, macrophages, and it forms the granuloma like a structure. So this bacilli remains inside this macrophage as well as the is this granuloma like a structure. So it's limit their growth and multiplication. So it develop the latent kind of disease. And what are the characteristic of the active state bacilli or the active stage of disease, particularly if you talk about the Mycobacterium tuberculosis? In active state, it has a replicating, it will be a replicating bacilli. It stain positively with acid fast staining and higher metabolic rate, and it's include either drug sensitive, multi-drug resistance, or extreme drug resistance bacilli. So either kind of bacteria will be developed the active disease, while this latent bacilli remains non-replicating state inside this granuloma or the inside the macrophage. It loses acid fastness property and has a lower metabolic rate, obviously, because this latent it's in a non-replicating state, so it has a low metabolic rate, and this is recognized as a phenotypic drug resistance characteristic. Please note that this drug resistance is not like as MDR or XDR, because this is a genotypic drug resistance, while in this case this bacilli is not responding to any drug because they are not multiplying, and that's why the drug is not killing to them, and it's recognized as a phenotypic drug resistance, totally drug resistance. So what are the conditions responsible for this different causing active disease or the latent disease? Mostly in active disease, the aerobic condition of well oxygenated condition will be there, physiological pH will be there, and it has a nutrient rich condition. While in granulomatic condition or the in latent case, there will be hypoxia because the surrounding the many cells and it's forming a granuloma. So the environment, microenvironment is like oxygen limited, nutrition limited, and developing the acidic pH, and how many kind of oxidative trace like as reactive oxygen species or reactive nitrogen species produced inside this phagosome. So it's come across to the various oxidative type of trace, and that condition are responsible to transfer this active bacilli to the non-replicating bacilli or the dormant stage. So based upon this condition, there are several dormancy model has been developed up to date, and mostly this in 1933 the scientist Lobel has developed this nutrient starvation model. What he did, he added this well-grown tubercular bacilli in the PBS, that just the phosphate vapor saline, and keep incubated up to the 1 month or 2 months, and he has seen that it's growing, it's maintaining its growth after growing on the new fresh media. So in a PBS, it's remain live, so that it's remain as a nutrient starvation model, but it's not become a well physiological model, because inside the body there will be a slight other sources will other carbon nitrogen source will be there, energy source will be there. So another model is there, that it's very famous model, when hypoxia. The hypoxia means just decrease of the oxygen condition. It's a gradually decrease the oxygen condition. If you look into this graph, so if the organism or the bacilli is growing actively, with aerobic condition. This is just a cotton plug and continuously aerating. So it has a higher metabolic rate as well as higher growth rate. And while if you keep the rubber stopper in that tube, it will be shows the some lower growth rate up to certain time, and after that it is becoming a flat, means it's becoming a bacteriostatic. And it's causes, it's the reason behind that the oxygen is depleted as the slower growth is there, whatever the present in this tube, and then it's become a bacteriostatic, so it's become a dormant. So it's resembling the whatever what is actually happening inside the host, particularly in this tube. However, there are some not some other conditioning media also used to modulate or to transfer this active population into the dormant population by using a such kind of multiple trace or vitamin C, that is AA is the vitamin C, ascorbic acid is used to modulating this media component, and that creates some kind of oxygen limitation stress or the other biochemicals oxidative stress, and finally that bacteria is go in the dormant stage. So but this is also not that kind of relevance to the host, because this ascorbic acid or other multiple stress like carbon monoxide or that kind of stress is not occurring in the host. So this model is considered as a best model, and using this model the drug screening assay has been developed, and this is widely used for the drug screening purpose. While the drug is added at this point, not at this point, because if you add the drug at starting point, it will be inhibit to the multiplying bacteria, not to the bacteriostatic or actual dormant population. So drug is added at this point, and to add drug here in this tube, we need a syringe or the which is loading with the drug, and we need to pass through this rubber stopper, so that the it will not break hypoxia and oxygen will be not mixed. So we need to maintain hypoxia condition in throughout this model, and this is a major difficulty, and it's a low throughput technique, because you need a large volume tube as well as syringe and all the large kind of material. So it's become a low throughput. So if you screen a drug in large quantity or large scale, it's not fit with the high throughput screening. So what is the high throughput drug screening program, or how it is developed? So well, you need to have first growth detection system. It's largely first development is done in at the low throughput, means in the tube level. And I think this microbiologist is well known with this detection system, that optical density or CFU used to detect this bacteria present in this broth. So you will, if you treat with this drug in the tube, you will get optical density or the CFU, means colony forming unit. So you will be check the treatment effect finally. But the problem again, that measuring optical density is not suitable for if dead population is there, and CFU measuring a CFU requires again large material like as plates, agar, medium, and dilution solution, and all that kinds. This technique is going to the low throughput. So researcher has developed many of dyes, which is I think already known in microbiological field, that is a resurgerin dye. Many fluorescence protein, luminescence enzymes are used to detect this bacteria. So if you add such kind of dye in this tube, this dye will be reduced by this bacterial population and will develop a color. So development of this color will be equal or the in proportional to the whatever the bacteria present in this tube. So by using this marker or this dye system, the proportionate amount of this bacteria are considered present in this tube after treatment or before treatment. So you will be easily calculate the percent inhibition by using this marker. So far, after developing this kind of the low throughput assay in the tube, the system is transferred to the large scale or the high throughput scale. This is just transforming towards the 96 well plate or the 384 well plate. What happens actually here, you will be decrease the cost expenditure, because you will require near about 10 times or 10 to 100 times quantity of the media or less material, like as here, if you are using a 4 ml, 3 to 4 ml, you will be require only in a case of 384 well plate, you will require only 30 microliter to 50 microliter volume maximum. Or in a case of 96 well plate, you will require a 100 to 200 microliter volume. So you will be reducing the cost expenditure as well as the drug dye, everything is reduced. And this we can do in a duplicate, triplicate, or chordate, whatever the multiplexing we can do, and at a time we can screen a multiple compound at a time, and we can measure. The beauty of this plate is that we can measure the fluorescence or the dye color through by using this plate reader. So it will give exactly the that's atom absorption maxima. You will be read the plate and get the final quantity of the bacterial present in this tube after treatment or before treatment, and final calculation is done, and the high throughput program run in this way in a particular drug discovery. So I will talk about just about the general drug discovery, why it's high throughput is necessary. Because if you look total program of general drug discovery, it will be take near about 15 years, almost 15 years, 10 to 15 years is needed to develop one kind of drug. And if you want to one drug, you need to start with the 5,000, at least 5,000 to 10,000. I think it should be a more than 10,000 compound. Then you will get a good drug which will be in a market. So starting with this large number of compound, you need to have a high throughput screening model. So this step, particularly drug discovery step, is done in wet lab, means laboratory level. Then it is after getting some hit molecule, like as supposed to 250 molecule will be get as active against this in vitro assay or wholesale assay. Then is subjected to in vitro toxicity or in vivo toxicity, admit profile, that is absorption, metabolism of that compound in vivo, because many of this compound either it is inhibiting at this stage, but that kind of this compound will be metabolized in the in vivo system, so it's not beneficial to us. So that compound is rejected at this stage, and finally in vivo efficacy is determined by in vivo TB anti-TB model, and we will get a few number of compound, like as 3 to 5 compound, and that is finally tested in a clinical trial, that is phase 1, phase 2, or phase 3. So it will require near about 6 to 7 years again, and after completing the phase 3, then it will go to the regulatory approval, and finally it's come in the market. You will see this a large time consuming a process, but need to go any drug discovery procedure need to go through this stage. Otherwise the it's not possible to get one kind of good drug in the market through this drug discovery program. So for this, what kind of assay available in the this laboratory level in the tuberculosis drug discovery program is, if you focus on that particularly in against the active stage of the tuberculosis disease, there are many or lots of assay available, because it's not need any modifying any condition, or it will be in a general condition, it will be regulated on, you will get a compound or the target hit molecule easily. So for this against the dormant state, there are very limited assay available, and whatever the assay is available here, which is also having some complication, which I will be discussing next step. And this at the intracellular level, there is also very less, means a very low assay, or the very less number of the assay available, and also in vivo efficacy testing, that is anti-TB testing in at the in vivo level, there is very less. Means there is only one model is available current date, and that is also have some drawback. I will also talk about that in my next slide. So if you look about this assay has also some complication, and the particularly this assay is also based upon the hypoxia phenomenon of the dormancy. So people have developed hypoxia in this, either using a tube or the plate, just 96 well plate is a seal, or the putting this plate in this anaerobic chamber. So hypoxia will be developed, and mycobacteria will be transferred in after some time, means after the 7 to 10 days, growing at anaerobic condition or the limited oxygen condition, it will be transferred to the dormant. But the problem is that addition of the drug, how we will be add the drug. In this situation, it will be possible that you will be add through the syringe, but it is a low throughput, and here you need to reopen this thing. And this is a actually problem of addition a drug in this at a particular dormant state and recognizing the dormant inhibitor. Otherwise, this model are showing the active state inhibitor, not a dormant state inhibitor. So I think every microbiologist know about this. Paraffin is heavy oil and reported to generate anaerobic condition in bacterial culture. So we have take this we taken this advantage of this paraffin because it's generating anaerobic condition, and we just top up this media with the paraffin layer and find out that it's gradually decrease the oxygen level if mycobacteria will be grow under the paraffin or overlaying with the paraffin. And now it's easy to dip the syringe or the tip just tip through this paraffin layer and insert the drug without the breaking the hypoxia condition. So smartly we just transfer this tube level assay to the microplate level. But then again, another limitation that in this plate we are unable to measure growth or the detection system, because we top up with this paraffin oil and it's limiting the growth detection system. So we have generated this red fluorescence strain by transforming the M cherry plasmid, which is containing fluorescent gene. And finally, we detected whether there is a paraffin top up or overlaying of the paraffin is becoming a difficulty, or is becoming a hindrance to the measuring of growth. But no doubt there is it's not complicated and it's not making any hindrance during a measurement of the growth at the 100 microliter of paraffin oil we use in or overlaying in the plate. So we used this model and just to check out this some standard anti-TB drug. And if you see this first line or the first line anti-TB drug, within that the rifampicin is supposed to the positive control of the dormant TB, and it's showing the growth inhibitory action against the dormant stage as well as the active state. But the rest of this drug is not showing the good inhibitory activity. MIC value is increased near about, in a case of isoniazid, it's near about 100 to 10,000 times. So you can see the risk of the drug resistance, that particularly phenotypic drug resistance in a case of isoniazid. And I think the nowadays the government program is included this isoniazid in the treatment for the drug for TB disease, and it's not effective. It's clearly indicate that it's not effective against the latent infection or the drug resistance population. If person is carrying the asymptomatic infection, it's not inhibiting the dormant population. So using this frontline drug is not useful to treat the dormant population, and we need a new drug against this to treat this dormant population or the two latent tuberculosis diseases. So so this model nowadays we are using in our laboratory and screening a more compound to identify the dormant stage inhibitor. A study is going on. What about the future of this in vivo dormant model? Well, we have the dormancy in vitro dormancy model, but what about the in vivo model? Because there is only one model available, that is a Cornell model. It's well recognized. What actually they did in this model, that they inoculated higher population of AM tuberculosis in this mice, and they after inoculation they incubated some time to develop symptoms, and after that they started isoniazid and pyrazinamide treatment. So they are well-known cell wall synthesis inhibitor and active against the active stage of the disease. So after treatment there is no disease, or there is no symptoms. Maybe this bacteria are totally treated or the totally killed, and they have determined by the CFU. So this because there is no CFU, it means that there is no disease. And so they keep this mice further in a sterile state up to the 4 week, and they found that there is no symptoms. But a further incubation of that mice or the keeping the at a sterile stage, this mice develop some active TB disease. How this bacilli is arising here or is developing the TB disease, although there is it is maintaining in the sterile state, because this drug is only inhibiting to the active stage of this bacilli, and the latent stage remain as it is, and after some time you will get this dormant TB. So far this model is used for drug screening purpose against the dormant TB, where they inject the drug at this stage. So after getting a read out the this active stage of the disease, compound is inoculated at this stage. So it probably they are assuming that it will be inhibit to this population, dormant, already present dormant population. But believe me, this is not inhibiting the actually dormant population. Whatever the disease is relapsing at this stage, it's inhibiting to the again active stage of population, not actual dormant stage, residing the dormant stage population, because within 8 weeks it start multiplying and the drug is acting against the multiplying stage. So we need to have such a detection system, because almostly this dormant bacteria also have another characteristic, that it is a non-culturable, it shows the non-culturable phenotype. It means that it is unable to grow on ordinary media, and that's why it's not give any kind of CFU on the plate. So we need to have a good detection system, like as a RFU, means red fluorescence or fluorescence kind of system, to detect this bacilli under the door, and particularly against the dormant stage. So the efforts are going in our laboratory. I'm actually purposefully not showing this data, that is not published yet. And so we need also the in vivo anti-TB dormant model to screen a drug after getting, if you get the good active molecule in vitro, you need to test in vivo efficacy in vivo. That's why we also need this in vivo dormant TB model. So the effort are continuing. So let's talk about the some COVID infection risk in the latent tuberculosis, because this latent tuberculosis is reactivated at any time or any immunosuppressive condition, and this TB and COVID-19 infection are both respiratory tract infection. So the infection site is similar, as well as TB resides for longer time in a patient body. Thus, it is assumed that there could be a greater on a greater risk for COVID-19 infection. And this is a one report also tell that the TB also get a severe COVID-19 infection with having a new symptoms like as acute cough, fever, and shortness of the breath. This is the shortness of the breath, also symptoms of the COVID-19, but due to this co-combined infection, it's giving a acute cough and severe condition, and they are on greater risk. So far, one of the unpublished clinical investigation also support this hypothesis that the TB means already infected TB patient are on a greater risk for the SARS-2 COVID-2 virus, that is a similar strain of the COVID-19. And it's possibly they find out that these patients are also have a severe symptom of this COVID-19. So it's supporting the hypothesis that the already infected TB patient are higher on a risk of COVID-19 infection. But there will be also second thing is that the latent TB can be reactivated anytime. So upon infection of this COVID-19, so this will be make a immune suppressive like a condition, and again this TB, which whatever the latent TB is residing in the patient body, will be reactivated, and it simultaneously develop both kind of symptoms. That is, one is active TB symptoms as well as the COVID-19 symptoms. So these patients are found to be the greater risk on greater risk on the diseases, both kind of diseases. So, or it's assumed that the BCG vaccine is also effective in the COVID-19, and is it truly actually effective in the COVID-19? Let's talk about that thing also. If you look about this BCG vaccination program worldwide, this as I already talked that the European and North American countries are lessly infected with the TB, and also they have not BCG vaccination program, but the rest of the world have BCG vaccination program except Australia and New Zealand. But and this in this country, in early phase of the COVID infection is not find out. Means in early phase there is a very low rate of COVID infection. So people or researchers are thinking that the BCG is there, BCG vaccine is there in this country, so that is the reason the COVID is not spreading in this country. But the current report is not supporting to this hypothesis, because this COVID-19 infection is spread throughout the world, and you can find now these days is severe in the most of the Middle East, Asia, or the as well as Russia, which is getting most BCG vaccine, as well as Brazil. So it's not recognizing it's not depend upon the BCG vaccination, and it's spreading throughout the world. But nevertheless, this data actually just I pick up from the today's newspaper, New York Times, and it's still that even though the Brazil, Russia, and India has a very low death rate, if you compare with this UK, Spain, or European countries, or United States, which is not getting a BCG vaccine, they have highest death rate of due to this COVID-19 infection or coronavirus infection. So it's ultimately tell that these countries are higher risk, or probable hypothesis we can put, these countries are higher risk or showing a death rate due to the BCG vaccine, maybe or due to the BCG vaccine. And post to that, what is the principle behind that the BCG vaccination program, and how could it read out this overcome with this coronavirus infection? So if you see this, the BCG could lead to the antigen independent activation of bystander T cell or B cell from the monocyte, and it gives the heterogeneous immunity, or it is termed as the heterogeneous immunity. Likely it's just a BCG infected to the macrophage, and it's just secrete the many inflammatory factors and activate the T cell or B cell. And the cell mediated immunity, once activated, it gives the immune protection against the another kind of disease, and it's also recognizing the another diseases like as a tumor. The BCG is also effective in the tumor, giving a immunity against the tumor also, because the T cell and B cells are cell mediated immunity is activated by secreting through this kind of factor. So it is thought that this will be a good mechanism, and by using such BCG vaccine, I think there are as per available current data, there are 10 BCG vaccination clinical trial going on in a various countries throughout the world against the COVID-19, and they are showing some positive effect of for a prevention of the severe COVID-19 infection among these older or healthcare patients, or sorry, healthcare people, not a patient. And this positive effects may be forced to us the giving a BCG vaccine. I think our well-known BCG vaccine producing company Serum is also doing some clinical trial in Mumbai region by using this BCG vaccine. And I think, but the result is not, they have not declared the result. So we are waiting for that result also. But the still question remains there, that how long does this heterogeneous immunity engendered by the BCG vaccine against this COVID infection? Because this monocyte will be lost long up to the month, and what will be the optimal time? Because this coronavirus is spreading, it has not limit, still it's spreading and spreading. So after 1 month, possibly if you give just the BCG vaccine to that person, it will be 1 month will be okay. After that 1 month, then again it will be infected with this COVID-19. So what will be the time limit or optimal time to vaccinate with this BCG vaccine is remain a question. So the study still going on. And then again the question is that what is the infection spread window in a primary affected countries? So when we will get rid out this through the outbreak. So if you just, I'm just mentioning that, or clearing that this is just a particular hypothesis based upon the available data, current data. If you look out this primary affected country, or the whenever the first time infection spread in particularly in European country, those countries are showing a minimum 2-month window, that higher peak means they are infected in showing the high spread infection in early March, and it's peak about in mid-April or the April month, and then it's decline. And so it's become a flat up to the in a May of month. So it's showing the 2-month window period in this primary affected country. And nowadays they don't see the many new cases in their countries. Might be these countries have 2-month window period. So is this possible, or is the same happening with this our country or the India or other rest of the country where the infection lately started spreading? Means in earlier March or the April, in India there was no spreading of that much kind of infection. If you see the 5,000 cases at this European country or at the India or the Brazil, see, it's lately started. So I think we can hypothesize that it will be require more 1 month time or the 20 days to flat down this curve, or the just new infection will be decreased after some time by completing a 2-month period. For the support of this hypothesis, the US is also started the early infection, but it's not totally flat. And I found out that particular reason, I found out the particular reason behind that. If you see this is a New York plot, means spreading the COVID-19 infection in New York. It's showing the totally flat within this after 2 months, but it's not flat with the in another state, because the US is a large demographic area. So state by state, it's showing a different kind of infection spread window, and it's possibly depend upon the demographic 2-month window. That's why this number is increasing. Still, it's going decreasing, but average number is still it's not totally flat. So maybe a demographic COVID-19 maybe have a 2-month window at a particular demographic area. So assuming that maybe will we require more days, some days need to wait some days to decrease our new case arising. But what will be the probable hypothesis why it's decreasing after 2 months? There is one hypothesis is put that the virus reaches as a virus reach at the higher population, it's also has higher rate of mutation, and so that the possibly different kind of variant will be arise, and maybe it lose the virulence, or it might be have some mild virulence, and the more virulent strain will be show the symptoms, while mild virulent strain remains asymptomatic, and other virulent strain will be 100% remain asymptomatic, or this mild virulent strain will be develop symptoms after some time. So based upon this data, and current data is also showing that there is a decreased death rate versus the active new cases. So it's also tell that the COVID-19 current COVID-19 infection is not becoming that much symptomatic, and it's not causing that much disease, maybe due to the arising high mutation rate in the person to person body, or next transfer, or just transmission of this infection to the next person. So due to this high mutation rate, arising high mutation rate, we will be get read out from the corona outbreak, maybe before coming the actual vaccine in the market. And probable hypothesis is that the high mutation rate is occur because this pathogen also need to adapt the body system or body physiology for their survival, and that's why it changes its genome, and actually it's causing means it's developing a more mutation in their genome, and it's decreasing its virulence, and finally maybe that's the reason death rate is decreasing. And another possibility is that it's also supposed that asymptomatic patient will be develop some symptoms, and they are also supposed to dangerous, but these asymptomatic patient will be spread the lower dose of this coronavirus, means although they remains asymptomatic, means that they have low population of the virus, or the low number of the viral quantity or viral particle in their body, so they will be just transmit the small number of the virus particle in the air or in the next person. So ultimately, this lower dose, I think, this is a lower dose is the another virulence property of the microbe, and at the lower dose might be a competition will be developed in the body, and person body will be develop good immunity against that virus at a lower dose. And this is just assumption that we will be get read out throughout this mechanism ultimately before having a vaccine or any kind of drug. So this is my just last slide, and it will be talk about just a safety precaution during the handling our common area or during handling of the material. So this is a just published report in the New England Journal of Medicine, and they talk about this. They measure the half-life time of the SARS virus, is similar strain to the COVID-19, and they find out that this virus remains up to the 2 hours in the aerosol or in a copper metal. So in aerosol, it will be sustained about the 2 hours, while it on the cupboard or the stainless steel or a plastic material, it will be sustained about 4 hours to 8 hours. So I think after 8 hours, it will get vanished totally, and we need not to worry about after 8 hours. So if we carrying any material or from the market or any from outsource, we need just to keep it outside the room or at a safe place, so nobody will be keep in contact with that material, and the infection spread will be ultimately going to stop. You need not to use any kind of disinfector also, because that is also ultimately becoming a toxicity. So what is my take home message through this the series of webinar? That is there is a need of appropriate diagnostics test for latent TB, and also need of in vivo anti-TB drug testing model to find out the new drug against the latent to fight against the latent TB infection. Latent TB can be reactivated at any time under the favorable condition, and so the latent TB patient could be on the higher risk of COVID-19 co-infection, as well as vice versa. Maybe a COVID-19 latent TB will be reactivated, or the COVID-19 infection, both are on the risk. And the BCG vaccine could be effective in COVID-19, but daily increase in COVID-19 cases raise the question on its effectiveness, and we need to optimize its time of vaccination. COVID-19 related strain could vanish within 2 to 3 hours in air, and within 8 to 10 hours on a metal or plastic surface. So it will be helpful to manage our common area or the surface, these materials. Although there are several clinical trial ongoing for COVID-19, like as a drug, chemists are trying to find out the best drug or the vaccine. Microbiologists are trying to find out the vaccine, but the still effort going on, and no prominent drug is available. So I will just request that use a mask in common area, make social distance, and take care of old age person, particularly TB patient, and stay safe. So with this take home message, I will acknowledge because this some of the part of work I have done in National Chemical Laboratory, which is and registered with the ACSR, and I have getting a funding from this Inspire body, DST Inspire. So with acknowledging these bodies, I'm very thankful to inviting community, inviting resources to this webinar, and thank you so much. Any question?
Thank you, Dr. Amar, for your informative talk in a very lucid manner. Now we are going to turn to the discussion session. For that, I will call upon Dr. Jaisingh Patil to take over. Dr. Jaisingh, sir.
Thank you, sir. Thank you, Amorevare, for giving us such an informative talk. Now, there are actually many more questions that have been asked by the participants. And on the behalf of participants, I would like to ask only few questions. So, Amar, the first question for you is, what is the extensively drug-resistant tuberculosis? The question is asked by Syed Azhar sir. What is the extensively drug-resistant tuberculosis?
Well, this when there will be resistance against this MTB resistance against the rifampicin and isoniazid, or that's for frontline DC drug, then it is recognized as a multi-drug resistance. But if more than that, like as pyrazinamide or this ethanbutyl also showing the resistance against that strain, then it is recognized as a extreme drug resistance strain.
Okay. Okay, thank you, Amar. Now, the second question is, some participants wants to know about the information regarding TB and relation with corona. What is the relationship between tuberculosis and corona, in short?
So, well, corona is a virus and TB is the mycobacteria. So this bacterium and the COVID-19 has a same site of the infection site, like as it's both are going to infect the lung, and they are developing somehow similar symptoms. Means COVID-19 have a similar symptoms, that is a cough and fever, and also TB gets the cough and fever, but TB is a longer time of it's a chronic infection, while this corona infection will be just for 14 days. So this is a particular difference in between this two infection, but the similarity is that they have both showing the same kind of symptoms and at a same infection site. And particularly this if you're looking at a BCG vaccination program, there is the cell mediated, particularly this is not a specific immunity, but yes, cell mediated immunity, that is also termed as the heterogeneous immunity, is giving a protection against the COVID-19, and some I think there are some report that's showing the positive result.
Okay, thank you, Amar. The next question is, what is the current status for latent tuberculosis? Have they identified any gene which is responsible for latent tuberculosis? Question asked by Samrina Bakal.
Okay, well, the particularly the gene is not responsible for the transforming to the active stage to the latent stage, but the condition are such kind of condition are there, like as a hypoxia, nutrient limitation condition, acidic pH. That's why this bacteria is transforming into the dormant stage, and ultimately it's shutting down many expression of the gene, as well as some of the gene which is required for survival. They have identified, like as some nitrite reductase or some DOS regulon, that is a dormancy regulated gene, are identified that which are upregulated during the dormant stage, and could be become a new target in this dormant stage, and still effort is going on that aspect.
Okay, thank you, Amar. The next question is, can't we made edible vaccine for tuberculosis? Because you have talked about the attenuated vaccines only.
Okay, well, actually developing the vaccine against this tuberculosis disease is a major hurdle, because there is no recognized particularly single epitope from this bacteria or virulence factor, and this has a particularly lipid or cord factor that is actually a virulence factor required for this TB infection, and against that it's very difficult to make such a kind of edible vaccine or any other kind of vaccine. So people are not interested to develop a vaccine, instead of they are interested to target with the good drug.
Okay, what are the strategies can be used to control immigrant TB? Immigrant TB. Actually, this question is asked by our MSc student Shailesh Hardkar.
Okay, immigrant TB, I suppose to not get all out this question. Immigrant TB means as per my knowledge, that immigrant TB, he supposed that from any other countries to us, or in demographic to demographic area. So in that respect, any patient will be have this dormant kind of infection. Any person, not a patient, because we don't know, we also are a carrier of this latent TB infection, and might on the base upon this in a favorable condition, that TB or immune suppressive condition, that carrier person will be develop a symptomatic TB, no
Okay,
doubt. So
okay,
in
okay.
immigrant cases, that is also developed.
okay. Thank you, Amar. This is the last question for you. What is the difference in symptoms of tuberculosis and COVID-19? The question is asked by Suresh Nikhalja, sir.
I think I have already explained this question, that symptoms difference.
Please elaborate it again little.
Actually, there is a difference, is that this fever is common in both the diseases, as well as the cough is common in both the diseases. But in the case of the tuberculosis, there will be a mucus containing a blood. So blood containing mucosa will be there, then it will be a TB. And if in the case of COVID-19, there will be no blood in your mucus, and sputum will be not there. Second thing.
Okay.
So it's
Okay.
very difficult to people are just getting a trouble from detecting a bacilli from sputum and getting a sputum from the patient. So it's very difficult to get a sputum. Actually, people supposed to that is a mucosa, but it's not actually a sputum. So if you get blood in the mucus, then it will be a TB.
Okay. Thank you very much, Amar.
Okay, thank you, Dr. Amar. This might have cleared the queries by and large in the minds of the participants. With this, I hope that the first session is over. Now we are going to turn to the next session, which will be conducted by Dr. Vaibhav Mahindarkar, who has joined us from Württemberg, Germany. For that, I request Dr. N.R. Sheikh sir to introduce Dr. Vaibhav in brief. Sheikh sir, please.
This is the second session we are beginning, whose speaker is Dr. Vaibhav Mahindarkar from Württemberg, Germany. He actually has completed his bachelor's degree in microbiology from Shivaji University, India. But for master's degree in microbiology, he had been student of our college, Yeshwantrao Chavan College of Science, Karad, which is also affiliated to Shivaji University, Kolhapur, India. It was in 2008. Since then, he got involved in research work. So for research, when he continued in Germany, he received his PhD degree in biology and biotechnology from Ruhr University, Bochum, Germany. During his career, he worked as a project assistant at National Chemical Laboratory, Pune, in India. Then he also has experience as an independent researcher at Department of Biotechnology, Lokmangal Biotechnology College, India. He was Senior Research Fellow at National Institute of Virology, Pune, in India. The institute, which is seen nowadays very much valuable for this COVID-19 pandemic. Then he joined for his research in 2013 at Institute of Biochemistry and Pathobiochemistry, Ruhr University, Bochum, Germany, where he was awarded with his PhD degree. Then he has technical expertise in molecular biological techniques like molecular cloning, heterogeneous expression of proteins in bacteria as well as in yeasts, protein purification. In short, he has technical expertise in protein studies. He has skills of IT, then skills to speak and work fluently in English as well as in German language, in Marathi, Hindi as well. During his career, he had some fellowships at his credit, like in 2011 to 13, Senior Research Fellowship from Indian Council of Medical Research in India. Then again, in 2013, Collaborative Research Center Graduate School Scholarship from Ruhr University, Bochum, Germany. So he started his career from 2013 in Germany. Then in 17 to 2018 WS scholarship from Gesellschaft der Ruhr University, Bochum, Germany. Although he is basically a microbiologist, he is now a protein biochemist with over 8 years of experience in protein biochemistry, molecular biology, and assay development, expertise in cloning, protein expression, purification, and characterization. He has some number of publications at his credit, out of which almost all are in international journals. He has some important work in virus research, which is the work he continued, I think, in National Institute of Virology. And now we are facing a problem of virus pandemic. So I think he also is a very right person for this international webinar. I welcome Dr. Vaibhav Mahindarkar. Thank you, Manrao.
Thank you, Dr. Sheikh, for the brief introduction of Dr. Vaibhav. Now, I request Dr. Vaibhav Mahindarkar for the next session. Dr. Vaibhav, sir, please.
Okay, so a very good morning to all of you. So I'm trying to share my screen now. So I hope you all can see my PPT and you can hear my voice. First of all, I would like to thank Professor Dr. Sheikh sir for the introduction. Initially, I wanted to present my work, which I did here in Germany during my PhD, which is also quite interesting. But as most of my research is unpublished in Germany, so I decided to present my drug discovery related research, which I have carried out at National Chemical Laboratory and National Institute of Virology. As coronavirus is a hot topic of discussion nowadays, in latter part, I will tell you about practical aspects of PCR and its use in diagnosis of coronavirus diseases. In National Chemical Laboratory, I was working on the project Purification and Characterization of cysteine protease inhibitors. Proteases are enzymes that cleave proteins. So there are different types of the proteases based on types of the amino acids which are present in their active sites. They are called as serine proteases, cysteine proteases, aspartic acid proteases, metalloproteases, threonine proteases, etc. Proteases are present in almost all living organisms, including bacteria, fungus, and even in viruses. So if we can control these proteases specifically, then we can control these organisms, and thereby we can control their pathogenesis. So there is a strong need to identify novel, specific, and potent protease inhibitors, which can be used as a drug molecule against the related diseases. The first part of the project was to establish a protease inhibitor assay. This was done by using a synthetic substrate, benzoyl arginine paranitroanilide. There is a protease cleavage site in between benzoyl arginine and paranitroanilide. So when protease action takes place, that will cleave this benzyl arginine paranitroanilide, and paranitroanilide will be released in the reaction mixture. So the appearance of paranitroanilide can be detected colorimetrically by measuring the yellow color at 410 nanometer. When we add inhibitors in this reaction, inhibitor will bind to protease, and this will form enzyme inhibitor complex. So the substrate will not be affected and it will not be cleaved. So there will not be any production of yellow color, and this decrease in the production is directly proportional to the protease inhibitor activity. In our search for novel protease inhibitors, we found one of the fungus which was isolated from mangrove plant. This fungus was showing 16 protease inhibitor activity in its extracellular culture. We identified this fungus as a penicillium species STBF1. This fungus was not showing any activity against serine aspartic acid proteases. So this fungus was producing specific 16 protease inhibitor. Then we did some time profile analysis to check when this 16 protease inhibitor is produced by the fungus. And we found that this fungus was producing 16 protease inhibitors from 4th day, and the inhibitor was active until the 7th day. So we harvested the extracellular culture of this fungus on 5th day. Before going for purification of this inhibitor, we also did some stability studies using different temperatures and different pH. And we found that this inhibitor is very stable in high temperatures, as well as it is very stable in different ranges of pH ranging from 2 to 10. Then we proceeded for purification of this inhibitor. Purification was carried out by step-by chromatographic methods. Firstly, we started purification by using ultrafiltration, in which we used firstly 30 kDa filter, then 10 kDa filter, and then 3 kDa filter. Our inhibitor was low molecular weight inhibitor. So by this way, we have avoided all the higher molecular weight impurities from our inhibitor. Then this filtrate was lyophilized so as to get the concentrated form of our inhibitor, and further purification was carried out by using gel exclusion chromatography. So in gel exclusion chromatography, we found that the inhibitory activity was concentrated in the 3rd peak. So we pulled this peak, we concentrated it using lyophilization, and again further purification was carried out by using reverse phase FPLC. Here FPLC is the fast protein liquid chromatography. We found our 16 protease inhibitor in this active fraction. So we pulled this fraction, again lyophilized it, and final purification was carried out by using reverse phase HPLC, where we use 5% to 95% acetonitrile gradient for the purification. Single peak on HPLC is indication of pure inhibitor. This inhibitor was showing IC50 value of 78 nanometer and Ki value of 97 nanometer. Kinetic studies showed that this inhibitor is competitively inhibiting protease. This research was published in the International Journal of Advanced Biotech Research. Additionally, we also found clostripain inhibitors. Clostripain is a 16 protease, and we also found this inhibitor. And this inhibitor can be used as a drug molecule against clostridial infections. And this research was published in Organic and Biomolecular Chemistry. After successful completion of my projects from National Chemical Laboratory, I joined National Institute of Virology. Here I worked on characterization of hepatitis E virus putative helicase protein. This characterization led us to give an idea about the sites of drug targets in this helicase protein. Hepatitis E virus is causative agent of hepatitis. It is mainly transmitted by fecal and oral route. It is mostly dangerous in pregnant women, where mortality rate is up to 30%. And no any drug molecule is available against this infection. This is the geographic distribution of hepatitis E virus. This shows how many countries are affected due to this hepatitis E virus infection. Hepatitis E virus is non-enveloped virus, which is approximately a size of 27 to 34 nanometer. This has a positive stranded, single-stranded RNA, and it is about 7.2 kb in length. Hepatitis E virus genome consists of 3 open reading frames: open reading frame 1, open reading frame 2, and ORF3. ORF3 codes for small phosphoproteins that are involved in regulation. ORF2 codes for structural proteins or capsid proteins. And I was interested in ORF1, which was coding for helicase protein. Helicases are motor proteins that use energy from ATP or NTPs, and they unwind duplex RNA or duplex DNA. So this is ORF1 of hepatitis E virus genome. It codes for methyltransferases, cysteine proteases, helicases, and RNA-dependent RNA polymerases. Hepatitis E virus helicase protein shows 7 signature motifs. Signature motifs are small repeating sequences, and these are considered to have some function. So our study was aimed to identify role of these motifs in helicase activity. For that purpose, in our lab, one of my senior have cloned wild-type hepatitis E virus helicase gene in PET vector. This is for expression of this protein into bacterial cells. This was done by Dr. Yogesh Karpe. We have deleted individual motifs from these wild-type helicase using site-directed mutagenesis. The next step was to express this protein in E. coli cell. This expression is carried out by using IPTG induction, which is on the basis of lac-operon principle. Further, proteins were purified by using affinity chromatography. There are two options to purify proteins by affinity chromatography. You can purify them by using under native conditions, or you can purify them under denaturing conditions. Our protein was little hydrophobic in nature, so it was accumulating in the inclusion bodies. So it was not possible to purify our protein or helicase protein by this under native conditions. So we purified protein in denaturing conditions. Renaturation was carried out by using renaturation buffer, which contains a little amount of detergent. Further purification was carried out by dialysis and gel exclusion chromatography. And here, this is the STS page of helicase protein and mutant proteins. So this is the band for helicase protein, and these are the mutant proteins. This is the Western blot analysis of the same proteins using anti-His antibody. As I said earlier, helicases shows NTPase activity and unwinding activity. So helicases use ATP as energy to unwind duplex RNAs. So this ATPase assay was already established in our lab. So we have established and optimized the quantitative assay for duplex RNA unwinding. For this reason, a duplex RNA was prepared by using in vitro transcription. And this one of the RNA was labeled with biotin, and other RNA was labeled with deoxygenin. Then we added this duplex RNA in the streptavidin coated plate. Streptavidin has affinity towards biotin, so one of the strand will be bound or kept attached to the 96-well plate, and other strand is free. Now we added anti-DIG antibodies coupled with horseradish peroxidase. This gives color after addition of substrate. When helicase reaction takes place, this results into release of the upper strand or DIG labeled strand, and no color is observed after this helicase reaction. And this reduction in color is directly proportional to the helicase activity. So we have compared percent NTPase activities and percent unwinding activities of all the mutant proteins with wild type helicase protein. And we found that deletion of motif 1A and deletion of motif 3 leads to significant loss of percent unwinding activity. And these are unique motifs which can be used as a potential targets for antiviral therapy. So these are some publications which we have achieved by using this research. In the next part of my presentation, as promised, I will explain the practical aspects of PCR. And in the last part, I will tell how this PCR can be used for diagnosis of coronavirus disease. This is a principle of PCR, where the first step of the PCR is denaturation, which is achieved by heating the sample at 94 degrees Celsius, in which the template DNAs are separated from each other. Next step is annealing, where DNA primers anneal to the template DNA, and the next step is extension. It is a polymerization step. So these three steps are repeated again and again, to be precise, for 30 times to achieve desired concentration of our particular fragment of DNA. These are the components involved in PCR reaction. It includes DNA template, dNTPs, buffer, primers, DNA polymerases, and in some cases, MgCl2. Ideally, a single DNA template is also sufficient for this PCR amplification, but we use about 50 to 500 nanogram of genomic DNA, and 50 picogram to 50 nanogram of plasmid DNA for such amplifications. The NTPs used are 50 micromolar to 200 micromolar. Then there are different buffers used depending upon the polymerase you are using in this PCR. Buffers contain different concentration of salt, MgCl2. We can also adjust these concentrations to have better results. We can also add some additives like DMSO, glycerol, PEG, Twin20 in this buffer so as to get better PCR results. These are some recommendations for primer designing. So these are very basic recommendations, such as primer length should be of 20 to 30 bases. Both the TM of both the primers should be similar. There should not be more than 4 bases in a row because this will reduce specificity. We should have at least 1 or 2 G or Cs at 3' end for better binding or elongation. And primers should not hybridize with each other or itself. If primers hybridize with each other, then they will be self-utilized and therefore they will not be available for actual polymerase reaction. This is a self-complementary calculator tool. Here we can see whether our primers are self-complementing with each other. So you can just Google for self-complementary calculator tools and you will get up to 10 to hundreds of such tools. You just have to paste your sequence in the tool and then you have to click on the check self-complementary and you'll come to know whether your primers are showing some self-complementation or self-annealing properties. You can also calculate TM in a similar way. You have to paste the sequence of your primer and just click on the TM, calculate TM, and you'll get the TM value. Then finally, important component of PCR is DNA polymerases. In our lab, we use 3 types of DNA polymerases: Taq polymerases, PWO polymerases, and PFU polymerases. Taq polymerase doesn't have proof reading facility, so it is more prone to errors, while PWO and PFU polymerases have this facility. In general, for amplification of short fragments, Taq polymerase is used. For amplification of larger fragments, PWO polymerase is used. And for amplification of very large fragments, such as inside-directed mutagenesis, where we amplify complete plasmid, in that case, PFU polymerase is used. And this is the actual thermocycler. There is initial denaturation for about 2 minutes. It is generally for hot start reaction. Then there is a denaturation for 15 seconds. This denaturation should not be so long. It should be only for 5 to 15 seconds. If we increase this temperature, then it might hamper the other PCR components such as NTPs. Then most important thing in PCR is annealing, adjusting annealing temperature. Annealing temperature should be 5 degrees Celsius lower than the TM of the primer. This is not a rule, but it generally works like this. So you can change this annealing temperature and check which annealing temperature is better for your desired piece of DNA. And this annealing temperature should not be longer than elongation temperature also. Finally, the extension or elongation. So we do this extension at 72 degrees Celsius for Taq polymerases. And for PWO polymerase or for PFU polymerase, we do this elongation at 68 degrees Celsius. And there is a final extension for 5 minutes, and the PCR product is stored partially at 4 degrees Celsius. And if you want to store it for longer, then you can keep it at -20 degrees Celsius. So there are various variations of PCR, like real-time PCR, reverse transcription PCR, colony PCR, nested PCR, multiplex PCR. And each of these PCRs have their own advantages. So you can use these PCRs depending upon your needs. I will explain this real-time PCR in brief. Real-time PCR is used for measurement of DNA or for measurement of gene expression level. Difference between real-time PCR and normal PCR is that in real-time PCR, we can monitor amplification of DNA in real time. This is done by using fluorescent dyes or DNA probes. So here you can see that during primer extension, cybergreen dye attaches to the newly formed duplex DNA. And then after attachment, it shows fluorescence. And this fluorescence is proportional to the presence of target DNA. So by this way, we can use this assay for detection or diagnosis of coronavirus RNA. In fact, RT-PCR is considered as a gold standard in diagnosis of COVID-19. Before going into the diagnosis part of COVID-19, I'll give you a short introduction about what coronavirus is. As we all know, coronaviruses are enveloped viruses with positive stranded RNA, and they are having size of about 120 nanometer. They are transmitted mainly through person to person or through droplets. And as of now, about 6.5 million cases have been reported, and about 3.8 lakhs of deaths have been reported until today. It has a high mortality rate in old people, and no drug and vaccine molecule is available by now. This is a geographic distribution of COVID-19. So here you can see that how many countries are affected due to this COVID-19 disease. On 11th March 2019, WHO declared this as a global pandemic. This is a structure of coronavirus. Here in red, there is a genomic RNA. It is a positive strand RNA, which is attached with this N, which is a nucleoprotein, which is also called as capsid protein. Then there are membrane proteins, envelope proteins, spikes. This is genome of coronavirus, and it has open reading frame 1A and open reading frame 1B. This codes for replication related proteins, and these S, E, M, these region codes for the structural proteins. Now I will explain in short how the replication of coronavirus takes place in our body. When the coronavirus comes in contact with our cells, it attaches to the ACE2 receptor of our cells. Then it is endocytosized into the cell. Then the positive stranded genomic RNA of the coronavirus is released into cytoplasm. Then translation of ORF1A and ORF1B takes place. This produces replication-related proteins and enzymes, including RNA-dependent RNA polymerases. This form a negative stranded RNA molecule. And this acts as a template for the generation of positive stranded RNA molecule or genomic RNA molecule. It also produces various subgenomic mRNAs by using discontinuous transcription method. So most of the envelope proteins or the membrane proteins, these are synthesized near ER. Genomic RNA is transported towards the ER where partial assembly takes place. From ER, these viral particles are transported to the Golgi complex by using vesicular trafficking. And from Golgi, these viruses are released outside by using endocytosis. Now we will focus towards the diagnostics for this COVID-19. There are basically two ways of, or two types or two methods of diagnosis. One is by using serological testing and other is by using RT-PCR. In market, there is a serological diagnostic kit available, and this kit is very easy to use and it's very quick. So you can get results in 10 minutes or so. So this kit consists of a sample buffer. In this sample buffer, we need to add about 2 to 3 drops of our blood or serum. And then this solution is then added on this sample pad. And there are 3 markers in this kit, M, G, and C. Here it is the same markers. M codes for, or M is for IgM antibody, G is for IgG antibody, and C is for control. So if you find only one line at the control level, then person is negative with coronavirus. If you are IgM positive, it indicates that the person is recently infected with coronavirus. If a person has IgG positive, it says that person might have recovered from this coronavirus disease. And if both IgM or IgGs are positive, then it says that person's body is still fighting with coronavirus, or there is a chance of secondary infection. Here is the principle of this method, where in this sample pad, we add the serum or blood of the patient. If the patient is positive with coronavirus, then this serum will contain antibodies against coronavirus, which will then migrate towards the absorption pad. In the conjugate pad, there are coronavirus antigens, which are attached with the gold nanoparticles. These gold nanoparticles are of size of about 100 nanometer, and they show purple color. So when coronavirus antibodies come into the conjugate pad, they form antigen, antibody, and nanoparticle complex. And here at IgM line and IgG line, IgM antibodies and IgG antibodies are captured respectively. This results into the accumulation of gold nanoparticles on this line, and this shows positive result. As I already told, this method is very easy and quick, but for this method to work, person or patient should get infection first, then produce antibodies against that infection, and then those antibodies are detected by using this serological test. So early diagnosis is not possible by using these tests. This can be easily explained by using this diagram. On the x-axis, you will find days, and on the y-axis, there are antibody titers. 0th day is a day when symptoms appear. IgM body starts to accumulate in the body after 0th day, and it reaches its peak on the 7th day. So by using these serological tests, we cannot identify whether patient is infected by this coronavirus in its initial 14 days. But by using PCR, we can identify whether or diagnose whether the patient is infected by this coronavirus in this 14 days period. And this is the major advantage of PCR, because if we diagnose this coronavirus infection in initial stages, then we can take further actions to prevent further proliferation of this virus. These are the factors affecting proper diagnosis. First is the time of sample. So if you are collecting sample at initial phases of diseases, then you should collect sample for RT-PCR. And if it is at the later stage of disease, then serological testing is the better way to go for. Then the next question is, what is better sample for RT-PCR? To answer this question, researchers took samples from nasal swabs, oral swabs, then urine, blood, and stool samples. Urine and blood showed they were always negative for viral RNA. Stool samples occasionally showed viral RNAs, and nasal swabs and oral swabs showed high viral quantity or high viral titer. So these samples are collected for RT-PCR based diagnosis. And the third important thing is transport of sample to the lab. This is a very important thing. Ideally, samples should be transported to the lab immediately as soon as possible. But if you are transporting this sample in the first 2 days, then we can keep these samples at 4 degrees Celsius. And if this transport is taking more than 2 days, then it is recommended to keep these samples at -70 degrees Celsius. Finally, testing and reporting. In 2020, January 2020, Korman et al. Have published one paper for detection of coronavirus by RT-PCR. In this paper, they have mentioned all the aspects of RT-PCR which are required to diagnose coronavirus. They have also mentioned which genes should be used to target for this RT-PCR analysis. Some of the researchers says that nucleocapsid gene or capsid genes are more specific and better targets for this RT-PCR based coronavirus diagnosis than that of the envelope genes or RNA-dependent RNA polymerase gene. But in principle, all these three genes works very well with RT-PCR. Sensitivity of RT-PCR is about 91%, and PPV, which is a positive prediction value of this RT-PCR, is 100%. This means that there are no any false positive results in RT-PCR. Finally, I would like to summarize everything. Firstly, we have to collect samples from nasal or oral swabs. Then there will be a transportation. Transportation is carried out at cold conditions. Then RNA is extracted in the lab. Then cDNA is prepared by using reverse transcription. RT-PCR was analyzed by using RDRP, E or nucleocapsid genes as the target genes. And if you found fluorescence above threshold level, then the test is considered as positive. And if you found this fluorescence below threshold level, then test is considered as a negative. As I already mentioned, this RT-PCR has a PPV value of 100%. That means if the person is showing positive results with RT-PCR, then 100% that person is infected with COVID-19 virus. Finally, I would like to acknowledge Dr. M.I. Khan and group from NCL, Dr. K.S. Lole and group from NIV. And I would also like to thank Dr. Kengar sir, Dr. Sheikh sir, Dr. Sawant sir, and Dr. Rao sir for giving me opportunity to present my work in this webinar. So, and finally, thank you all for your attention. And the topic is open for discussion now.
Thank you, Dr. Webo, for a very informative talk. Now, the topic is open for discussion. I call upon Dr. Avinash Raut to conduct this discussion session. Dr. Avinash.
Thank you, Dr. Yaga, sir. And Very nice informative session by Dr. Vaibhav Mendarkar. So the questions which are being asked by some of the students as well as some of the participants, which are maybe faculty members also. So first question from Samrina Bakal is that what are the causative symptoms that leads to high mortality rate in pregnant women?
Okay. As in hepatitis, actually we don't have any drug molecule or any vaccine in hepatitis. So if the person's immunity is low, then it is difficult for person to recover by their own. So for treatment for hepatitis is to just say, just stay home and maybe to take some ribavirin antibiotic. But for pregnant women, it is as their immunity is very low, it is not possible.
Thank you. Second question is knowing the morphological and physiological aspects of hepatitis E virus, why vaccine has not been invented? It is the question is by BD Sunil.
This is a very nice question. Actually, I don't know answer of this question. One of my friend is working on this preparation on this ORF2 protein, which codes for the capsid proteins, and they're trying to isolate this capsid protein and inject it into the mice to check whether they are developing some antibodies against this hepatitis E virus, but they have not found good results until now. But I don't know the exact reason of that.
Okay, well done, Vaibhav. Then the next question is by Siva Sankari MSD Murthy. Can we use hepatitis S vector for COVID-19 vaccine development?
What do you mean by hepatitis S vector?
Okay.
I'm sorry, but I didn't understand the question. Hepatitis E virus.
Okay, let's go to the next last question. Can you suggest best tool for primer designing?
Yeah. Actually, there are lots.
This question is by Wajiha Banu. Can you suggest best tool for primer designing?
So there are lots of programs for primer designing and for cloning, but the most important and simplest program I found was Snapchat. In this program, you can just click on the open reading frame, which you want to do, and then just there is a primers to these. So it's very simple. You just need to click on the go into that program and say add primers, and primers will be added automatically to that particular fragment of DNA. So I recommend you to use Snapchat tool.
Okay, thank you very much, Vaibhav. So I again request Raikar sir to continue.
Thank you, Dr. Avinash, for conducting this particular session. In such types of webinars or conferences, a number of questions are there, and due to the time constraint, it's not possible to answer all the questions. So we are now coming to the end of this webinar. I now request Professor T.B. Sawant sir, organizing secretary of this webinar, to propose the vote of thanks. Sawant sir, please.
Good morning to all. I am here to express my vote of thanks. Today, we have organized one-day international webinar on current perspectives of microbial diseases 2020. On behalf of our management, Sri Shivaji Education Society, Karad, Board for Higher Education, and our college, Yeshwantrao Chavan College of Science, Karad, I express my sincere thanks to Dr. Amar Evare, postdoc fellow from USA, New York, and alumni of our college, has given very nice information about the correlation of tuberculosis and COVID-19. And also, he explained awareness about the COVID-19. Once again, thank you, sir. Then after, I am also very much thankful to our another alumni, Dr. Vaibhav Mendarkar, a scientist from Germany, has given excellent information regarding the diagnostic aspects of COVID-19. Once again, I am very much thankful to all, to both of them. Then my sincere thanks to Honorable Balsab Patil, Minister of Cooperation and Marketing, Maharashtra State, and Chairman Sri Shivaji Education Society Karad, Board for Higher Education, and Honorable Prakash Pandurang Patil, Bapu, General Secretary Sri Shivaji Education Society Karad, Board for Higher Education, and other Management Council members for their expert guidance to this successful webinar. I am also thankful to Dr. B.N. Gopne, Coordinator, YB7 Competitive Exam Center, Karad. Again, my sincere thanks to our principal, Dr. S.P. Kenga, for his expert guidance and very good organization of this webinar. Again, my sincere thanks to Dr. N.R. Sheikh, convener of this webinar, and Dr. B.P. Redekar, coordinator, IQAC, for their expert efforts to success this webinar. Then after, I am also very much thankful to all the members of organizing committee and Dr. B.P. Redekar for anchoring this webinar. I am also thankful to coordinator of this webinar, Dr. Dev Patil, Dr. A. Rao, and Sri K. Sutar. Finally, I am very much thankful to all the participants and delegates of this webinar. Once again, thanks to all. With this, I declare that this webinar is over. Thank you.